much I shake, I'm a little earthquake. I'm bending at the knees, sir, catch me please. Don't wanna sleep, my heart's still beating, and maybe it's a virus.
So this is Julia Haltigan and the Hooligans with Maybe It's a Virus. Pretty catchy tune. I actually like it. And again, sadly, I can only play you just a little bit of it. However, there is a link on YouTube. So if you want to listen to the whole soundtrack, or the whole song, you can certainly do that. And she has a lot of interesting songs. I like her music. Okay, let's move into microbiology. I've divided this week's lecture into several parts. As I mentioned last week, we were going to finish the viruses this week, and really, we're only going to touch on basic viral characteristics. I wish we had time this week to go into the different herpes viruses and HPV and all those really great types of viruses, and we will, but I'm going to wait until the end of the semester when we talk about a lot of infectious diseases and disease causing organisms. So I'm rather restricted this week to just sticking with, again, basic characteristics of viruses and one really great example of a virus so that you have a, sort of a model, a representation of what viruses can do and how we can treat them. We're also going to talk about prions. The what? Hold on. And we're also going to talk about controlling microbes. Yay! And if I forget to say so, happy Halloween for the week. I know that's coming up on Tuesday, so yay. Here are the objectives. Again, a little tweaked. Again, we're bringing the viruses back. What I wanted to point out to you, if you have forgotten, I wanted to point out that the outline is, whoopsie, the outline is due November 5th, right? At 11:59 p.m. So get that attached in the Dropbox. And if you have any questions, just email me, okay? Okay. Also, our very first real quiz is going to be next week. So it's going to be big. All along, I've been giving you little mini practice quizzes, but this is the biggie. Okay, so that's going to be due November 12th, and you're going to have a quiz over cells, viruses, control, and host defenses. So, okay, be ready for that. All right. How about some terms for prions and viruses? As always, I am including these in your PowerPoint lecture notes. However, I wanted to let you know that I am placing the terms for control and control agents later on, so I've broken it down a little bit. This might have to be tweaked just a little bit too. Okay, I haven't double checked some of these. Also, you are responsible for knowing the function of the following antiviral drugs and why they do work on viruses. So that's coming up, by the way. Oops, sorry. All right, so I used a term, prion. Miss Eden, what's a prion? I'm so glad you asked. So prions, first of all, prions cause a condition, a syndrome known as spongiform encephalopathy. Gesundheit. Yeah, I know. All right, break this down. Spongiform, like a spongy form, like a sponge. Encephalopathy, like pathology. Encephala, like encephalitis, that's the brain. So maybe this causes a spongy brain pathology. There you go, spongiform encephalopathy, and that's what prions cause. Now, prion, where did they come up with this name? What does it really mean? Prions stand for proteinaceous infectious agents. So I underline the protein part and the infectious part because prions are proteins. But just proteins? Uh-huh. That means that they are made of strands of amino acids. And if you remember Mr. Seberg's lessons, that proteins sometimes have a three-dimensional shape, and for them to do their job, they have to keep their three-dimensional shape. Shape means function. So over here we have these coils of amino acids to make up a prion protein. So they're not living? No. Up till now, everything we've talked about, either, it consists of a single living cell or many different living cells, prokaryotic cells, eukaryotic cells. They have all the things that are cellular and characteristic of living things. No nucleic acid? No. No DNA, no RNA, no genes. Fascinating, huh? So what happens? Well, these are proteins, and again, they appear around the cells of the brains, the neurons, and they are bad, and they fold badly. They are misfolded. Because of that misfolding, they cause plaque formations in the brain. Now, if you're familiar with Alzheimer's, you know that, oh, that's kind of what happens with Alzheimer's too. Yeah, plaques in the brain, bad. So the proteins misfold, they form plaques in the brain. Now, the interesting thing is when you have a single misfolded protein, a prion, it will actually bump up to other proteins and bring them to the dark side and convert the normal proteins into bad proteins. So they become misfolded, they become prions, they become bad. So the prions bring healthy, good proteins to the dark side. Hopefully you're Star Wars fans. You get that. You get that reference, okay? If not, email me. Oh, here's a picture to the right. An autopsy. Brain tissue. Looks a little spongy? Yep, sure does. So the misfolded prion proteins accumulate in neural tissue, particularly in the brain, so central nervous system tissue. And this accumulation occurs slowly over long periods of time. This causes the brain neurons to die. The brain develops holes, sponge-like. Well, if you have holes in your brain function is going to deteriorate. So, the syndrome that we call transmissible spongiform encephalopathy. This is something that happens when you catch a prion. It's transmitted to you. Now, some individuals actually can make the misfolded prions just accidentally. We know that this does happen in some animals, but these prion particles can be transmitted from animal to animal, human to human, animal to human. Okay, so human disease progression. Over here we have two brains, by the way, the control, the normal on the right, and this is the brain that has a spongiform encephalopathy. You can't see the holes here, but you can see that the brain is shrunk and that it looks not very healthy. Okay, so human disease progression, depression, paranoia, aggression, all over tingling and numbing sensation, palsy, like the shaking. Okay. What typically occurs, especially towards the end of this patient's life, is pneumonia, because there are so many things going on. That patient is susceptible to a lot of other infectious disease causing organisms. So many viruses may move into the lungs and cause pneumonia, bacteria, and that's typically how a patient will die. So, different kinds of spongiform encephalopathies. Okay. So again, some of the symptoms that occur not just in humans but in all animals that get it is loss of motor control, dementia, paralysis, wasting away, eventually death, like I said, from pneumonia. The different types, again, we're going to talk about all the different kinds of spongiform encephalopathy. So the first type is bovine spongiform encephalopathy, or BSE. You might know it as mad cow disease. This first appeared in 1987 in Great Britain. So here you see a cow, and I won't show you the video, but if you go onto YouTube, you can see the downer cows and that they're falling down, they're stumbling. They're making sharp, jerky movements, and it's quite sad. And the muscle tissue starts to disintegrate, and they start to get kind of a wasting away. You can also see this in sheep, and yes, we see this actually in the United States, actually. So a condition known as scrapie. Okay. So here's scrapie, and the animal, again, think of these proteins are infecting the central nervous system, and the poor sheep is going crazy, basically. It's getting such a sensation. Think of an itch you cannot scratch, but it drives you crazy. The animal will rub its hide against anything, fences or trees, until it's raw or bleeding, and that animal must be destroyed. And I will talk about control with prions in just a little bit. It's a very difficult thing. Chronic wasting disease. Now, this you can also see in the United States. This is seen amongst mule deer and elk. I know a lot of hunters, they know about this, and so do butchers who will take care of your deer or elk. Some of them are misinformed and they will instantly cut off the head and get rid of the brain material, but you never cut off the head because we know that the prions can also be found in the central spinal fluid. The central nervous system fluids. So you never cut off the head because the fluids might wash over the other tissue. The best thing to do is to keep the central nervous system, brain, spinal cord intact, and then take out the pieces, that like hindquarters and places that are the best places to eat. Um, if you see an animal like this, of course, you would never, ever, ever, ever try to eat it. You would call some kind of animal control services and be very careful with this. Oh, sorry. One thing, let me back up just a little bit. So, there's a lot that we do not know about scrapie and chronic wasting disease and prions in general. Controlling prions, for example, is very difficult. A lot of the methods that we use to sterilize instruments and fluids actually won't work on a prion. We know that, for example, formaldehyde used in embalming fluid kills everything, which keeps basically the the person from deteriorating, the deceased from deteriorating. It kills fungi and bacteria and anything else that might cause decomposition. So formaldehydes are used. Formaldehydes actually make prions much more sticky and more infectious. We know that if you bury a human or an animal with some kind of spongiform encephalopathy, there will still be active prions years and years and years and years later, at least 10, 15 years, we know. We don't know beyond that because we haven't been studying it for very long. How about heating? Well, that's an option. But I will tell you that one of the reasons the mad cow disease spread through the cattle in Great Britain, let me move back just a little bit. All right. One of the practices that they used, and by the way, so did we, and we do also with other types of animals. But one practice is when you slaughter an animal, you take the best pieces for human consumption. So, for example, the cow, you would take out the pieces that would make steak and hamburger and anything else that would be good human food, and some of the other not so palatable pieces, maybe feed them to animals. And then you have a lot that nobody else wants. So that would include your central nervous system. So what they would do is they would take what's left of the cow, including the brain, and they would heat it into pellets. Think animal kibble, like Purina Chow. Yeah, turn it into that. So you take the cow, what's left of the cow, make little kibbles, and it's going to have a lot of proteins. And you feed it back to the cows. Yeah, making them carnivores. Yeah, not just that. they're actually cannibals too. They're eating themselves. Now, this was how the prions got into the herd. There was an infected cow, had prions. The prions were in the central nervous system. They ground up the brain with the rest of the material, made it into cow pellets, fed it into fed it to a whole bunch of cows. They became infected. And then, of course, their brain material was turned into kibbles. And this is how the mad cow disease spread throughout Great Britain. I'll mention a little bit if you've lived in Great Britain, traveled to Great Britain, had beef in Great Britain, you cannot donate blood products, or organs, okay? Because you could spread prions, spongiform encephalopathies. All right. So let's talk about humans. All right. So with humans, like I said, you get loss of motor control, dementia, paralysis, wasting, eventually death. The first case of human prion disease, spongiform encephalopathy, occurred actually over a century ago in a little village in Papua New Guinea. There is a cannibal tribe, and what happened was when somebody in their tribe passed away, they would not throw them into the earth. They thought that was horrible, so they would consume the recently departed. Okay, they would cannibalize them. And well, they would pretty much eat every part, and this is how it spread through this tribe in New Guinea. They no longer do this, and the cases of Kuru no longer. I mean, we it has dropped. Eventually, sometimes they do get a case, but it's very rare. All right, another modern name for prion disease in humans is Creutzfeldt-Jakob disease, CJD. You say you've heard of that. Good. I hope you have. If not, you're sitting there thinking, I've never heard of that, but I hope I don't get that. If you look to the right, here's some brain tissue. Here you have CJD and Kuru. Pretty much it's very similar. Here it's in cows, here it's in sheep, but sheep, both are spongiform encephalopathies. Now, when it comes to Creutzfeldt-Jakob disease, understand there are genetic forms. People just have genes that made proteins that misfolded. We see about 200 cases per year in the US, by the way, but there are transmissible forms, which really mostly stemmed from people of Great Britain who ate those infected cows, or people who ate the imported meat from Great Britain during that time. So again, we started to see these creep up in the 1990s. So they ate infected meat. There are, one other thing, there are some cases that we don't know about yet. People are still just starting to study these things. But some cases have creeped up, and we're not even sure just exactly where they got the spongiform encephalopathy. Some other people might get it from a tissue graft. So you had a surgery, somebody donated tissue to you, and you contracted spongiform encephalopathy that way. I know of several people who performed a surgical procedure. The physician, one of the surgical nurses, and surg tech all died because the patient had spongiform encephalopathy, CJD, and they were not protected enough, and they contracted CJD and died. So, well, how do I know I've got? Let's say I had I performed or assisted in a surgical procedure and found out later on that patient had Creutzfeldt-Jakob disease. And okay, how do we diagnose this? Maybe I've got it, maybe I don't. Unfortunately, diagnosis doesn't occur until symptoms appear, and I already mentioned what the symptoms were. And this might take up to 10 years, by the way. It's cumulative. We can do an autopsy, but of course, that's not going to be something you want. Treatment. We don't have any drugs. There's nothing. The best we can do is be supportive. So, supportive medicine for that. Okay. So, time for a quiz. What's a prion? A virus containing DNA, a virus containing RNA, an infectious protein, a bacterial cell, a eukaryotic cell. You're right, it's just an infectious protein. It is not a virus. It is not any kind of cell, and it does not contain nucleic acid, either DNA or RNA. Good. Okay. What's a virus then? Over to the right, in red, you see Ebola viruses, and they are actually uh, coming out of an infected kidney cell. Okay, this is actually a monkey kidney cell, but the cell has manufactured and is now releasing viruses. I'll tell you up front, viruses are not living. They don't grow, they don't metabolize, they are manufactured within the host cell. I'll repeat that a little later. All right, first, okay, I'm watching. Raise your hand out there if you are infected with a virus right now. You sure? You should all have your hands raised at this point. Um, yeah, you're all infected with lots of viruses. You may not have any diseases, but yeah, you all are. You have viral nucleic acid inside most all of your cells. Probably not red blood cells because they don't have you don't have your nucleus in there. But most notably, you probably have several types of herpes viruses. So congratulations, you are all infected with viruses, lots of them. Okay, so how about some general characteristics? Okay, first of all, viruses are really small. Now your bacteria are just really small. But okay, add some reallys in there, and you have viruses. Now for your bacteria, if you remember, those are in the micrometer range. So down below in the lower right-hand corner, we have a animal cell like your cheek cell. Here's a frog egg here. Here's a cheek cell. And here's a bacterial cell. Of the cells usually can be measured with micrometers. So if you took a quarter and you divided it up into 1,000 slices, one of those slices would be a micrometer. But what about a virus? So here is a flu virus. Well, we tend to measure viruses using nanometers. So if we could take this quarter and chop it up, slice it up into 1,million slices. Okay, one of those is a nanometer, so that's very small. Let's just go ahead to cellsalive.com, one of my all-time favorite websites. It's just so much fun. So let me see if I can enlarge this, make it a little larger. Okay. So they use an electron microscope, and this is something, of course, you can see with the naked eye. This is a meter that's going to, or a key that's going to show you how big things are. So from here to here is 20 millimeters. So millimeters we can see with the naked eye. All right, so let's move in. Okay. And here we're into about 2 millimeters. Here's a hair. Okay, what is this little thing here? Oh my, these are dust mites. Dust mites, very huge. Okay, here's the gray part. These strands are your hair. Here are the dust mites, and this is about 200 micrometers from one side to the next of this little key bar. Okay, micrometer. All right, what is this? There's something in there. Huh, looks nice, big and white. Let's see what that is. I think that's a pollen grain. Why, yes. Okay, so here we have this micrometer bar at 20 micrometers. The yellow is a pollen grain. You have a big white, you have this kind of purpley thing. That's a yeast cell, baker's yeast. You have a white blood cell here in the front, and you have red blood cells there. And then you have something else. Let's move in here. Ah, here we are. So the green guys, this would be, this is E. coli. The yellow guys that are circles, right? This is Staphylococcus. And this thing that looks like almost a needle with a little eye at the end, that's an Ebola virus, which is actually pretty darn huge. Uh, but wait a minute, you said that viruses are measured in micrometers. Yeah, but Ebola is a little bit of an exception. He's huge. So now let's look at the normal sized viruses. So for example, here we have rhinoviruses that cause the common cold. Shall we magnify these things a million times? Okay, here we go. So now we're at the nanometer range. Okay, cool? I think they're cool. Okay. Again, now when it comes to viruses, they have no metabolism. They don't eat. No, they don't take in food or oxygen or anything else. They don't even replicate themselves. They don't grow. They don't move. They're not motile. Not at all. Their job is to get inside a host cell. And let me tell you, if there is a cell, a real cell, bacterial, viral, plant, protozoan, fungal, there is probably a virus that will take it over and become part of it. And the virus will insert its genetic information, its genes, one way or the other. It could be kind of tricky. And the genes then will tell the cell to make more viruses. So it hijacks the cell's replication machinery. Now, virologists tend to classify viruses according to, well, the type of cell that they infect. So, for example, sometimes they infect eukaryotic cells or prokaryotic cells. Oh, sorry. Sometimes they infect, birds, sometimes pigs, sometimes humans, sometimes bats, sometimes cats and dogs. Sometimes they cross over and sometimes they do not. Now, if they live in a human, sometimes they're very fussy. They might live inside a human cell, but they only like a white blood cell, or they only like a liver cell, or they only like a respiratory epithelial cell, like a lung cell. And that's all they like. So that's how we classify them. So virus shapes. Okay, I'm only going to mention three. Yay, you say. The first one is cylindrical. So it's a cylinder-shaped virus. The nucleic acid is coiled up inside. This could be either DNA or RNA. A good example of this is this bullet-shaped virus, the picture taken with a transmission electron microscope. And it looks like a bullet, but it's helical. And this is the rabies virus, by the way. All right, the second kind of virus shape is icosahedral. The what? If you took some geometry, you might have learned that an icosahedron has a lot of triangles. It is an extremely efficient structure, very strong, doesn't even require a lot of resources. So over to the right, you see all these little tiny purple circles here. Can you see these? They look like they're stacked like, well, they look like my new tile from my new bathroom floor. But these are many, 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 many chikungunya viruses. Okay, causes a really nasty viral infection. Okay, if we have time, we'll talk about chikungunya. Okay, and then there are the weird shaped ones. Pleomorphic, if you remember that term, means many possible shapes depending on their mood. So here's influenza virus, and over to the right, Ebola. Okay, very, very weird, very pleomorphic. So how do viruses spread? How do they infect? Tell me about viral transmission. Okay. Well, as you have been working on your bacterial research project, you have come upon many different means of bacterial transmission. Well, viral transmission is actually very similar to bacterial transmission methods. Okay. So, okay, the first one, the biggie, fecal oral transmission. Well, that means that somehow fecal matter, and I like this. This is a nice little World Health Organization. And well, actually, I guess it's not World Health Organization, but they're trying to educate people that, okay, the types of material that come out of the colon, all right, they may look invisible when you wipe everything off, and they may not even stink, but the invisible particles are there, whether you went number two, okay, you defecated, or you're cleaning a diaper. So it's very important to wash your hands, okay, and be very careful before preparing food or eating or serving food or anything like that. So when people get a fecal oral transmitted virus, it's because, well, okay, you got it because you didn't wash your hands. The chef who made your food didn't wash their hands, which is why there's always a sign, employees must wash their hands. The chef cross-contaminated some foods during preparation. So maybe his hands weren't very clean, or he was using contaminated utensils, pans, pots, something like that. Maybe you're consuming produce that was contaminated. Maybe it was washed with contaminated water. Water had fecal material from someone's, maybe their um, from their cattle, or maybe there was some fecal material, municipal fecal material that got into your water source, into your well, or an animal just thought while he was grazing on your beautiful little garden that he would just, well, leave a little package for you. Okay. Flies. Flies are really good at hanging out on top of feces like this dog poo here, and then hopping on your potato salad. So flies are good at dropping off viruses, as well as bacteria and other microorganisms, by the way. And remember, these things are invisible. Can't smell them, can't see them. Okay. So yeah, she thinks everything's just, and I wanted to put this pretty picture because everything looks so clean, doesn't it? And safe. Yeah, that's when you get into trouble. All right, what else can transmit viruses? I'll tell you what, biting insects are really good at doing this, and mosquitoes are by far the biggest culprit. Now, we have a new term. When an infectious agent is spread by, say, a biting insect, then we call that biting insect a vector. So a vector spreads an infectious agent. Mosquito carries the viruses, bites you, and injects the virus into your tissue and bloodstream. Okay? But mosquitoes aren't the only ones that do this. But let me tell you, mosquitoes are good at spreading viruses and bacteria and parasitic worms and malaria and all kinds of things. So, viral transmission through respiratory droplets that float in the air. Now, they're quite invisible, and hey, I feel fine. So, why should I have to sneeze into my sleeve? Or I could just sneeze into my hand. I feel fine. Well, here's the thing about viruses and even some respiratory bacteria, but turns out that you can be feeling fine and transmit influenza and an awful lot of other viral respiratory diseases way before you even have a symptom. So, and if you are sneezing, well, all right, one sneeze is going to contain an awful lot of infectious viral particles. Now, little tidbit, by the way. Do you know what they say when they say that you are more likely to get influenza and colds, so respiratory viruses in the wintertime versus the summer? Why is that? Well, now a lot of people will argue that during the summertime, you're outdoors, you're not in small spaces, maybe your stress levels aren't as high. And that's true. That is all quite true. But there's a lot of research that strongly suggests that humidity levels will influence how well your viruses can infect your respiratory tract. How can that be? Well, in the summertime, if the viruses are loose, they could bump into humidity droplets. And then they combine, they become heavy, and they don't get into the respiratory tract as effectively. As opposed in the wintertime, it's dry and little invisible particles of viruses floating in droplets can travel great distances. Okay. Now, viral transmission can also occur because of sexual transmission. Okay. So here's the thing, condoms always lower the risk. And I've said it once, I'll say it 1,000 times. You do want to reduce the numbers. So, yes, condoms always a good idea, always lowers the risk. However, understand that there are some viruses, like the human papillomavirus, that you know it is HPV. It is transmitted very, very easily by mucous membrane to mucous membrane contact. So, skin contact. Okay, just saying. All right, so let's talk about some terms, virions versus viruses. What? Yeah. Okay. So let's define what is a virion and what's the difference between a virion and a virus. Okay. So when I sneeze, heck, okay, the actual infectious particle is floating around in the air. That is the virion, the actual infectious particle. Now, a virus is a little more broad than that. It is true that this could be considered a virus and viral structure. However, once the little virion infects my cell, here we are on the right, and that's a eukaryotic cell, right? And the virus gets into my cell, and it might break apart, but the viral DNA then might incorporate into my DNA. Now, this is throughout the whole process, this is a virus. It doesn't have to be the actual infectious particle. So it could just be the DNA that is now inserted into my nucleus. Okay? So the virus, we could just be talking about viral DNA hiding in your chromosomes. That's still the virus, which is again why you are all infected with viruses. Okay, maybe not virions, but viruses, yeah. Okay, so that is an arrow showing this little squiggly the part. This is supposed to be the viral DNA in brown, kind of a brown orange. Okay, all right. So let's look at the virion infectious particle. What does that entail, you ask? I'm so glad you asked. So first of all, in green, we have the capsid. The capsids are protein little shells, and they are, so it's like a protein coat, and they are designed to protect the nucleic acid. Now, here's the deal. Viruses can be DNA viruses or RNA viruses. Why do we care? You got to find out that DNA viruses tend not to mutate quite as easily, and it's a little easier to get a vaccine against a DNA virus as opposed to an RNA virus. RNA viruses would include HIV and the common cold virus. Okay. This is not always true, but this is why we want to know, is it a DNA or RNA virus? Okay. Then virions, in order to be infectious, sometimes they have to carry their own specialized little enzymes, and that will help them infect their new host cell. So, because these enzymes are very unique, it might be a very good antiviral target. Well, you can't attack nucleic acid. That would be bad because your cells are loaded with DNA and RNA. So, yeah, that might be tricky. Protein coats, yeah, that's not going to really work. So, for a lot of antivirals, this is what we attack. Not always, but this is the best way to attack to not hurt the human cell. Okay. Some viruses are non-enveloped or naked viruses, like this picture to the right. Okay, so basically you have the green capsid, you have the DNA or RNA all coiled up inside. That would be the orange part. And that's pretty much it. Now, the thing about the non-envelope viruses, when they look like this, they tend to be stronger in the environment and they can last for a very long time. There's something called a norovirus that can ruin a cruise by causing, well, severe diarrhea and stomach cramps, and it can remain on surfaces for very long periods of time. Now, there are also enveloped viruses that we haven't seen before. So they get these envelopes, or the viruses get the envelopes when they leave the host. So the host will manufacture the virus capsid and all the nucleic acid. And then as the virus leaves, it will wrap itself up in the phospholipid bilayer, the envelope, okay, of the host cell. Now, it doesn't have to be the outer plasma membrane, but many times that is what happens. So the virus comes from the host that made them. Now, envelopes. Why have an envelope? Well, the envelope does protect the virus when it gets into the new host. The new host might try to send out digestive enzymes to beat up on the proteins, but the capsid green proteins are protected by that outer envelope. So it's an outer protective layer. Also, that outer membrane hides the virus from the immune cells. The immune cells are looking for those ID tags. Are you foreign? Are you foreign? And they might see the virus as foreign if it weren't for the envelope. That's not always true, but sometimes it is. But here's the cost. And sorry, I guess that's misspelled, but the envelope. If a virus that's supposed to have an envelope loses its envelope, then they can't infect anymore. So enveloped viruses tend to be very wimpy in the environment. So here's a little bit of trivia for you. HIV viruses. Nasty, huh? Something you definitely don't want ever, and nor do you want anyone else to be hurt by them. They are enveloped. Now, if you go to one of the research laboratories, like the one at Creighton, run by Michael Belshan, he will tell you, and he works with HIV viruses, and he says, if we spill HIV on the counter, it's only going to last seconds to minutes. Dries out, and it's no longer infective at all. So, yeah, seconds to minutes. But understand, now HIV, once it gets into the body, all bets are off. It gets into the body, gets into the cells, and it is very good at what it does. Sadly, there's very few things that are better at causing human diseases than HIV. And actually, we do kind of consider it the king. Okay, so some other general characteristics. Now, a lot of viruses use some structure and sometimes several structures to attach to the host cell. If you remember, viruses, they don't move, they accidentally bump into their host cell, and there has to be some receptor that latches onto them. So, some viruses latch on using a capsid. So, they use their green protein capsid to attach to the new host cell. Some viruses use envelopes, and sometimes with the envelopes, they'll also use these little things that are called spikes. And you see these are little orange spikes sticking out. Sometimes we call those papillomeres, and they use those to attach to the new host cell. So, okay, viruses somehow land on and attach to our cells, host cells. So, down below, you can see, all right, that kind of looks like a eukaryotic cell. And I said, let's pretend that's maybe a white blood cell. And here's our virus. So, remember, they don't move, they're not motile. They have big numbers, though, and somehow they have to land on and attach to the appropriate cell. And if the landing gear doesn't match up virus to host cell receptor, that virus doesn't get in. So, what are the steps? How do viruses get into our cells and take them over? Glad you asked. All right. So, and I'm sorry, I have some pictures here. They're kind of lame, but hopefully it still helps. All right. So the first thing that has to happen when a virus, is going to invade an animal host cell, first of all, is attachment. A fancy word for attachment is adsorption. So it has to attach to the host cell. Moving to the picture to the right. Okay, you can see here is a virus. You have that kind of blue capsid and the black nucleic acid. Maybe it's DNA. And again, the blue-green kind of capsid, and then it looks like there's an envelope. And oh my, there are spikes or papillomeres. And oh, it looks like it is attached to the host cell. How is it attached? Oh, by those purple little host cell receptors. Okay, and it's a perfect lock and key fit. So the host cell, like I said, has to have the right or correct receptors. And again, the virus can attach by its spikes, maybe spikes and envelope, if it's an envelope virus, or it might attach with just the capsid, or it might use a combination. And it's a random process, like I said. They don't wiggle towards the cell and the receptor. Okay, the next part is the whole virus has to get in to the cell, and really the most important part is getting the genome, the viral genome, that's the genes, DNA or RNA, okay, has to get in. Now, that can happen by many different mechanisms, but if you remember, cells sometimes bring stuff in by endocytosis. Now, bacteria don't do this, but our white blood cell sure does. And so there's a fusion, you can see, of the host plasma membrane with the viral envelope, and the receptors are also anchoring it, and the virus then can enter. And you have the capsid and the nucleus, or the capsid, and you also have the DNA or RNA. Now, it's all about the genetic information, right? The genetic information has to tell the host cell what to do, how to run its manufacturing processes so that more viruses can be made. Well, that means that the nucleic acid, DNA or RNA, has to be released out of that protein shell. So that process is called uncoating, the removal of the protein coat and release of the DNA or RNA. Interestingly enough, a lot of times the host cell, our cell, will send over enzymes that help the process, that degrade the enzyme capsid. So how unfair is that? So I know, okay, not the best picture, but ... This is what's left of the capsid, so that's the uncoating. All right, so now the genetic information has entered our cell. Now what? Okay, so it's in. The next part is biosynthesis, where it replicates inside one of our cells. So again, this could be our white blood cell. Okay, so the virus, using its genetic information, has to tell the cells and the factories of the cells to make viral nucleic acid and viral capsids and anything else the virus needs. So again, this is biosynthesis, the replication of the viral subunits. And now the method is going to depend on the type of nucleic acid. Because, well, just think about it. If it enters and the virus is a DNA virus, then it has to get its little hiney into the nucleus right away, because that's where all the DNA stuff is processed there. If it's RNA, then something really strange has to happen, because our cells don't listen to RNA, they listen to DNA and transcribe and translate that, just like Mr. Seberg said. Sometimes, again, certain enzymes have to be used in biosynthesis. I will use an example with HIV a little bit later on. Then the subunits come together, and this is the reassembly of the viruses. Now, there's no quality assurance department. There's no virus checking the other, or the production of all the other viruses. No, it's the reassembly of viruses is rather sloppy. So it's not an efficient process. And there are many empty capsids that didn't even get DNA put into it. But don't worry, they have their numbers. The last part is the release of the viruses. So, if it's an enveloped virus, it gently buds out. However, some viruses explode out of the host cell. Over to the right, you can see this is using an electron microscope, and these cells, I know they look very strange, these white, that looks like they're scaffolding. This is just showing you the cell skeleton. And you can see the big old holes. And the holes are where the viruses burst out of the host cell. So this could very easily be one of your nose cells, one of your respiratory epithelial cells. And let's say when somebody sneezed at you, achoo, they didn't cover their mouth. You inhaled the virus, it got in. There's attachment or absorption, it went in. It was uncoated, and then you get biosynthesis. And then when it releases, well, cold viruses do burst out. So one virus went in, and about 200,000 cold viruses come out. And that allows those viruses to infect more of your respiratory cells. Or you can sneeze this out at other people. So, okay, I know, awful. What are the consequences of a viral invasion? So I said the cell might burst, but that's just one of the consequences, and we'll come back to that. So, what happens to the cell during the invasion process? Well, the cells usually are pretty messed up when they're taken over by a virus. We call the result of a viral infection a cytopathic effect. This is something visual. If we want to abbreviate cytopathic effect, we say CPE, or the CPEs. Over to the right, you see these healthy little cells with a normal little nucleus. Okay, these look like they could be the types of cells that line maybe your cervix, and they are. But believe it or not, these things that are stained really, really purple, and you're trying to figure out if that huge round circle is a nucleus, which it is. All right, these are infected cells with HPV viruses, and they look messed up. So that's a cytopathic effect. So the cells look weird. This is because as the cells start to manufacture their virions, they do this in the biosynthesis stage, of course. Subunits accumulate in the cytoplasm, making the cell look grainy. So look how grainy that looks. Yeah, even this one looks a little messed up already. Now, the cells sometimes will fuse, and you'll get big, giant, multinucleated cells. So one cell just fuses right into another. Yeah, it's really weird. And again, these are called giant cells. What else can happen to the cell during invasion? Well, like I said, some viruses, they replicate, they get huge numbers, and they just break out of the cell. Okay, they lyse out, and yeah, that pretty much kills the host cell. Cold viruses do that. This is just somebody's 3D image, JPEG image, but it looks like the cells, in fact, did burst open, and you can see all the little viruses, the new virions, coming out. And this would be something that would happen with the common cold. Now, some viruses remain in the cell, and then they slowly, gently bud out, or they might leave by a type of exocytosis. Okay, so this is actually something that happens with HIV. Over to the right, you can see this is a blue white blood cell called a CD4 white blood cell. And all the little yellow spots are HIV viruses, and they bud out. And that will cause a persistent or chronic condition. Now, symptoms may or may not be immediately evident, but eventually they probably are. And again, my example, like to the right, HIV. I know, don't want to see a picture of a baby there, but it's just the way it is. Okay. What happens to the cell during the invasion processes when the virus remains hidden? Shh, quiet. Another name for this is latent. What does that mean? That means they get into the cell and they don't even replicate. They don't cause disease, they don't replicate, they don't do anything. They just hide. Okay? All right. So when that happens, lots of things can happen. Sometimes they're reactivated. Sometimes while the viral genetic material is hiding in the cell, the virus DNA can transform the host cell genetically. So that means it gets into our cell, it inserts its genes, and that messes with our genes. That's what happens with HPV. It gets in, it transforms our cells. By transform, we're talking genetically changes, okay? So transformed cells can contain mutations that mess up the cell's normal cell cycle. When that happens, cells will start replicating uncontrollably quickly and they stop listening to cell signals. They don't act like they are part of the body anymore. They start to act quite independently. They even mutate independently. Groups of these cells, if they clump together, can produce tumors. Now, tumors can be just benign growths, okay? So this would be, for example, plain old seed warts, or if they're on your feet, they are plantar warts. Warts are caused by viruses, period. Warts are caused by viruses. I can get that out. Yeah. Had a virus, or excuse me, had a wart ever? That was caused by a virus. You got a viral infection. Yes, you did. Okay. And believe it or not, the viruses that cause warts are HPV viruses. So they can cause seed warts, plantar's warts, or genital warts. Usually the strains that really transform our cells and produce cancer tend to be a little different though, but all of which is caused by HPV. So again, cancer can form and cells, when they become so weird and so resistant to other cell signals, they break free and they can spread to tissue. So this is as I've been talking about, uh, this is HPV that has been infecting cervical epithelial cells up above. Down below, you can see how the cervix is being swabbed, but it is showing cancerous growths here. Okay, well, tumors. And the next step is chemo, okay? Surgery and chemo. Now, something else that viruses can do, and many of these do, we're just starting to understand the processes, but many viruses cause immunosuppression, leading to diseases caused by other infectious agents, HIV being the biggie. Now, maybe you knew or maybe you didn't know that HIV infects white blood cells and then they can't fight infection. So somebody with HIV develops AIDS and they get all kinds of infectious diseases. Okay, so you're immunosuppressed. But there are other viruses out there that do exactly the same. They use different methods and they don't always infect white blood cells, but they suppress the host immune system. The other viruses that I am referring to would include influenza and measles. So both of those viruses, they get in, and if that's not bad enough, you can get another infection. Let's stop for a question. Some viruses like herpes remain in a cell but are completely silent until activated. These viruses are called what? Acute, chronic, latent, retro, or lytic? Latent. Latent was the word that we used. Yes. Okay. So, what virus cycle do we understand fairly well and can use as an animal virus example? Well, I'll tell you what, anything we understand well means that we had a lot of money to spend on research. There's a lot of money that was given to researchers who studied HIV. That's in the past and in the present. And thank goodness, because now we not only understand HIV, we understand a lot of viruses. So I am going to talk about HIV in detail. Hopefully, this will not only teach you about the HIV virus specifically, but it will also give you an idea of how viruses how they interact with our cells, and also how we develop antivirals. And by the way, there's some really pretty pictures on the right, well, a pretty picture on the lower right-hand side. There's a glass blower who likes to make viruses out of glass. So I think that's beautiful. Okay, so let's talk about HIV. HIV, a retrovirus. And over here on the upper right-hand corner, this is the actual transmission electron microscope picture. And here are a couple cartoons that I will refer to you to. Now, what does HIV stand for? HIV stands for Human Immunodeficiency Virus. Okay, AIDS is not a virus. AIDS is a syndrome. Okay, HIV is the name of the actual virus, this thing. Okay, but it's called a retrovirus. Why is it called a retrovirus? When you think retro, at least I do, I think of retro fashions, going back in time, kind of in reverse. Well, here's the thing. HIV goes in reverse, but it goes in reverse because it replicates its genes in reverse. So what? Okay, well, you see, HIV doesn't carry DNA, it carries RNA into the host cell. Over here, here we have the whole virion, and here we have double-stranded RNA. Now, if this gets into our cell, who's going to pay attention to this, to RNA? Nobody. Our cells pay attention to DNA, right? Okay, so HIV somehow has to make DNA from what it's carrying, and that's RNA. Now, cells don't normally, so what it has to do is, okay, it has to go in reverse transcription, right? You're thinking, okay, I kind of remember that word. Mr. Seberg mentioned that. Do you remember way back when you learned about how genes are made into proteins? We said that genes, the gene in DNA, okay, think of the nucleus of your cell. And in your nucleus, you have a chromosome, and on your chromosome, you have genes. So when a gene is going to produce a protein, that gene is transcribed. Okay, so it's DNA transcribed into messenger RNA. And then messenger RNA gets translated into proteins. Okay, so this is going not from DNA to RNA. We have to go from RNA to DNA. Well, you're going to need some special enzymes for that. Okay, so we will get into that. So morphology includes, okay, its envelope. So in gray, again, you see that it has a phospholipid bilayer envelope, which makes it wimpy on a counter, doesn't it? Right? Pretty easily killed on a countertop or anything else. Okay, again, it's an RNA virus, double-stranded RNA, and it attaches by special little spikes called glycoproteins or GPs. You don't need to know which one. There's like a glycoprotein 41 and a few others. So that particular little glycoprotein spike attaches directly to one of our white blood cells, a white blood cell that has a receptor known as a CD4 receptor. So over to the right, you can see, this little spike, this is the glycoprotein, is going to attach to this red CD4 receptor. So any white blood cell that has CD4 receptors are known as CD4 cells. Okay, consequently, we say HIV infects CD4 white blood cells. Anything else? Not as far as we know. Okay, now because it's such a weird virus and has to replicate in reverse, it has 3 very unique enzymes. Okay, so the first enzyme is called reverse transcriptase. It is an all-purpose polymerase. So here, okay, we have a reverse transcriptase. It's that bright blue little glob there, okay, right there, okay. There's also something called an integrase. Now, integrase, its job is to allow the viral DNA intermediate strands to be spliced in or incorporated into the host cell chromosome. So its DNA gets put into our DNA, using an enzyme called integrase. The last enzyme is called protease. The protease enzyme just cuts new viral proteins so the virus can be released. So you can see up here these little HIV viruses in the upper right-hand corner. They are being released, but there's a lot of these little guys in the cytoplasm of the poor little white blood cells. So before they can bud out, they have to have that little protein cut by an enzyme called protease. Okay, now something you don't need to know, but there are two subfamilies of HIV, HIV-1 and HIV-2. But what you might want to be aware of is that these types of HIV viruses are different genetically and biologically. So if you have a patient that comes in and says, "Well, my partner and I are both HIV positive, so we don't worry about protected sex." Well, here's the problem, and it won't probably be up to you to tell them this, but HIV-1 is by far the more aggressive virus and causes a more aggressive disease. So if I have HIV-2 and my partner has HIV-1, okay, that could make my situation even worse. So it's just something to think about. Another thing, though, that I do want you to know is that in one infected person, there's tremendous genetic variability. So even if I have HIV-2, I can have so many different strains, just tremendous strains. And if I did have catch HIV-1, I could have multiple strains of both. And now, mind you, again, if my partner is HIV positive as well, but my partner might have more antiviral resistance. My partner's viruses might be more resistant to antivirals. So yeah, there's more tremendous variability in when it comes to HIV viruses. The faster anything replicates, the faster it can mutate and give us great challenges in care, treatment, and prevention. Nothing replicates and mutates faster than HIV. Nothing, not even influenza. So, whoops. Okay, so I'm going to go through the stages of HIV replication, and this is also going to tie in those very special enzymes that I talked about. So again, it's a retrovirus. Now I know looking at this, it's a little busy, so I'm going to break it down a little bit. So stay with me. All right, so the first part of HIV replication is attachment or adsorption. All right, well, we already talked about that. So here's our HIV virus, and it has that little glycoprotein spike, and it attaches to a CD4 receptor, or, well, just one of the receptors. There are other ones there. Okay, like I said, the spike is a glycoprotein. You don't need to know that it's glycoprotein 41, but it's very specific and it only attaches to CD4 receptor proteins on white blood cells. And here we are. So it attaches to the white blood cells. Will it attach to another type of cell? No, it has to have this receptor. Once the virus has attached to the CD4 receptor, it is brought in by, well, something called receptor-mediated endocytosis. Our cells actually think that this is something good that it's supposed to bring in, and it does. So the spikes, the glycoprotein spikes, attach to the CD4 receptor and a couple others, and the envelope of the virus fuses with our plasma membrane, and everything is brought in. And here, the next step is viral uncoating, right? Well, because the RNA has to be released. Okay, so the RNA is loose. Now, once the RNA is released, well then, our cells are not going to pay attention to any RNA. So the HIV virus now is going to use reverse transcriptase and convert RNA to double-stranded DNA. But mind you, these are viral genes. Now, the next step is the viral DNA has to enter the nucleus. Well, because this is where our DNA is held, right? So we've got our chromosomes there. The second enzyme, called integrase, there we are, integrates the viral DNA in blue into our host chromosome. How are we once an HIV virus DNA gets integrated into our chromosomes, how do we get it out of there? No, we don't. It's in our chromosome, in our white blood cell. Yeah, tricky. Now, once the viral DNA is there, it is transcribed and translated just like all the other genes of this, that are on our chromosomes. And some of the viral, DNA some of the genes are used to make viral particles. Some of the viral DNA is used to make more RNA. Okay, in this case, viral RNA. And and some viral messenger RNA is transcribed and or translated at the ribosome to make a whole bunch of viral proteins. Okay, and the viral proteins then produce the capsid. And all of this, by the way, the capsid is produced in the factories, and the HIV RNA gets shoved into the capsid. Oh, sorry about that. And then the last part is release. So HIV, being an enveloped virus, is going to capture some of the host membrane, and it's going to wrap around the capsid. And of course, the viral RNA is inside. So when all is done, you have a brand new virion. These white cells will continue manufacturing viruses, and they're going to have a hard time doing anything else. In fact, they don't, it's hard for them to do anything else. Okay, back to that transmission electron micrograph picture. Here's the inside of the poor white blood cell known as a CD4 cell. If you remember anything about white blood cells, these would be like your big time T lymphocytes. Some of your major players are little, are CD4 cells. And look at this cell. How is it going to do anything but make HIV viruses? Poor white blood cell. It's in bad shape. You don't need to know this, but I just wanted to show you how CD4 cells do so much work for us. They trigger macrophages, and they even help B cells make antibodies, which is something that maybe you never learned or didn't learn well or forgot. So I will get back to this later on in the semester. So the white blood cells, they're all infected, and this is going to cause a syndrome, lots and lots of symptoms associated with an HIV infection. Believe it or not, the immune system does recognize that HIV is there, and it works very hard fighting these infected cells, trying to stop them. It's just that eventually it loses. Almost always. Not always, but usually. So again, white blood cells, WBCs like T lymphocytes become depleted. The patient progresses towards immunodeficiency. And this is something, and again, you don't need to really memorize this, but I just wanted to show you that you get these big drops in your CD4 white blood cell count. Now remember, your white blood cells are your soldiers. So without those, you are not going to function. You're going to be immunodeficient. Even though the body puts up a good fight, sometimes, it could take anywhere from 5-10 years, but it depends. It depends on the type of virus. Once you are immunodeficient, then you have a syndrome called AIDS. What does AIDS stand for? Acquired immunodeficiency syndrome. Some people develop immunodeficiency syndrome, but they got it genetically. This, you got it through a virus. You acquired it through a virus, so it's acquired immunodeficiency syndrome. AIDS. What causes AIDS? HIV. Okay, don't mix the two. AIDS is a syndrome, not a virus. HIV is the virus. So the transmission of HIV, yeah, mostly sexual contact, right? But also contaminated blood. It's a bloodborne pathogen. So blood and blood products. Um, Most tragic of all, of course, is that it can be passed in utero through the placenta into the infected mothers. Okay, or excuse me, from the infected mothers into the children, and children are born with HIV. Okay. Symptoms: swollen lymph nodes, weight loss, fatigue, dementia, and something called Kaposi's sarcoma. What happens, and Kaposi's sarcoma is actually caused by a herpes virus, human herpes virus 8, which will come up a little bit later. It's a cancer on the skin. We probably get exposed to this all the time, but we never get Kaposi's sarcoma. Why? Because I hope we're not immunodeficient. So a good white blood cell count would keep this from happening. Okay. Our white blood cell soldiers do lots of work. But if a patient comes in with Kaposi's sarcoma and, and probably some other symptoms of having a lot of opportunistic pathogens, fungal infections, uh, just lots of infections that people normally don't get. So the physician will probably do a test and look for HIV. Prevention, of course, monitor blood for contamination. We do a pretty good job of that these days. Safer sex. You know that there's no such thing as safe sex. Abstinence. If you're, if it's possible. Um, about casual. Oh, sorry. So what about casual contact? Okay. So if someone has HIV, AIDS. Are you going to get HIV viruses from casual contact? Maybe eating utensils, mugs, even probably a toothbrush. You think? What do you think? All right, you're not liking the risk, but remember this is enveloped on a counter, clothing, mug, eating utensils, probably even a toothbrush, although there might be, blood cells on the toothbrush, so I probably would not do a toothbrush. But casual contact, yeah, the virus is not going to survive in the environment. But again, once it gets into the body, into the white blood cell, all bets are off. Nothing, when it comes to infectious disease-causing organisms and their impact on this planet, HIV makes the biggest impact. It causes the most deaths, I should say. Okay? Treatment, antivirals. What can we do? The early antivirals were pretty darn toxic. Remember, this guy gets into our cells. So, and it's, the HIV RNA becomes DNA that becomes part of our chromosome. See this? So if it's housed inside of our chromosome in our nucleus, how are we going to get at it? Well, the early antivirals included something called nucleic acid analogs. Okay, now stop. Nucleic acid. Oh, like DNA and RNA are made up of nucleic acids. Okay. Analogs means sort of the same. Okay. So, all right, explain this. Okay, nucleic acid analogs are chemicals that look like the subunits of DNA but are not. Now, there's a little more to this, but just bear with me. I'm just going to keep it simple. Consequently, when those chemicals get inside an infected cell and inside the infected cell's nucleus, they will cause mutations and stop viral replication and all DNA from replicating properly. Would that not be toxic to the good cells, the ones that haven't been infected yet? Yep. So it would, if once it's in the cell, once it's in the body, it'll enter all kinds of cells. The cells that replicate the most will replicate their DNA the most, and they will be mutated and hurt and affected the most. So these kinds of drugs, okay, if you're thinking, then why do it? Well, here's the thing. Viruses replicate so quickly, and the DNA is replicated so very, very, very quickly, they will be hurt more than our DNA, our healthy DNA. But yes, when we have cells that replicate quickly, like hair cells, gastrointestinal epithelial cells, they replicate about every 3 days, 2 to 3 days. So yes, they're going to be hurt the most by these kinds of drugs. The big example of this is AZT. If you've ever seen the Dallas Buyers Club, okay, lots of, that is really a tragic story. But the toxicity of this drug is extraordinary because, again, it not only hurts the cells that are invaded with HIV, they're going to hurt all the cells. Every time a cell wants to replicate, it has to replicate its DNA. How does it work? AZT looks like thymine. Okay, so it even kind of has a thymine in there. It's actually thymidine, but we're not going to go there. Okay, I'm going to keep it simple. So this drug will base pair with adenine. So you have adenine bases with thymine. Remember, A's with T's and G's with C's. You remember the base pairing. Okay, so the drug AZT gets in there and kind of looks like thymine, but isn't quite. It pairs with adenine, and that will cause a mutation. Okay? Yeah, but you're right. It gets into all the cells, so lots of toxicity. Yeah, not good. How about some other ideas? Aren't there specific enzymes that HIV carries that are so very unique? Yes, couldn't those be targets? Oh, you bet. So first of all, reverse transcriptase. Our DNA never has to go. Our RNA never goes from RNA to DNA. We go in regular transcription order, okay? So we don't have a reverse transcriptase enzyme, but HIV has to have this. So there are antivirals that attack reverse transcriptase called reverse transcriptase inhibitors. How about protease? Yeah, we don't have a protease that, like this. So it's not going to really mess with our cells. Integrase, again, this is something HIV uses to sneak its DNA into our DNA. So this is a good antiviral target. Normally, because these viruses mutate so rapidly, and they do, they get around all of these drugs, all of them. So usually what doctors will prescribe is a combination of nucleic acid analogs, if not AZT, ones like it, as well as integrase inhibitors and some reverse transcriptase inhibitors as well, but usually combinations occur. The viruses still can mutate in such a way genetically that they can get around even these drugs and become resistant. Vaccine? Well, here's the problem. The HIV virus mutates too rapidly within the host and throughout the human population. A vaccine has to target one major ID tag found on all the HIV viruses. So if it's going to work, okay, you have to have one vaccine that has one ID tag that is found on all HIV viruses. But, well, they mutate too much. Okay? So vaccine has to target an antigenic characteristic found on all the viruses. Yeah, so that's not going to happen. Not in my lifetime. I doubt it. I can't imagine. We know a lot about HIV, and what we know is that, yeah, it's in that respect, it's pretty unstoppable. Let's do some questions. Yay! Okay. These HIV viruses will acquire a phospholipid bilayer and become enveloped. At which step did they acquire the envelope? Biosynthesis, attachment, budding out, reverse transcription, or integration? Right, they're budding out. Remember, they wrap themselves up in the host membrane. So as these HIV viruses leave the white CD4 cells, they carry some of the membrane with it. And that makes them basically protected inside the host, but wimpy outside. Why do retroviruses like HIV need reverse transcriptase? A, to escape from the host cell. B, to uncoat. C, to make messenger RNA from virus DNA. D, to make DNA from virus RNA. E, to attach to cells. Okay, retrovirus goes in reverse transcription. So that means we're going from, we have to make DNA from RNA. Okay, this allows HIV to make DNA, even though they only come in with RNA. Sorry, that was D. Why do retroviruses like HIV need integrase? A, for the virus to attach to the host cell. B, to uncoat. C, to make messenger RNA from virus DNA. D, to insert viral DNA into the host chromosome. E, to convert DNA to RNA. And of course, it's blocking integration of viral DNA into the host chromosome. So integrase D is there to insert viral DNA into the host chromosome to remain indefinitely for the life of the poor white blood cell. What type of cell does HIV infect? Oh. A, cells with the CD4 receptor. B, red blood cells. C, all white blood cells. D, liver cells with a glycoprotein receptor. E, all of the above. This one was tricky. It's A. Now, some of you probably said all white blood cells. Not all of your white blood cell soldiers have the CD4 receptor. Okay, so I was tricky here. But yes, white blood cells with the CD4 receptor. These cells with CD4 receptors are white blood cells. They're your soldiers. I was tricky on purpose. All right. AIDS is the result of the direct depletion of white blood cells, red blood cells, many types of cells throughout the body. A, white blood cells, the CD4 white blood cells. Yeah. Okay, to the right is a picture of an epithelial cell infected with the human papillomavirus. Yes, HPV. So which is true? This contains a virion. This contains a virus. This looks normal. This contains proteinaceous infectious particles. This contains latent bacteria. Okay, let's eliminate the latent bacteria thing. Okay, this is not about bacteria. This is about viruses. They're very different. Okay, this contains proteinaceous infectious particles. That would be prions. No, we're not talking about neurons and prions here. So does it look normal? No. So it's between these two. Remember, the virion is the complete infectious particle. Okay, so this is actually a virus. When we look at the cell, once it's infected, we just consider it one big virus. Okay, so we don't see any virions, but we do see it's infected with viruses. Okay, there's the difference. Okay, are we ready to talk about controlling infectious organisms by physical and chemical means? No antibiotics yet, you guys. That'll come. We had a little bit of antibiotics, and we'll get there. Okay, so first little cartoon. I wish that bathrooms did come equipped with a little alarm like this that said, didn't wash hands. Yeah, that would be nice. Okay, here are some terms again, and these are related to control, physical and chemical. Okay, first of all, I should mention that when we talk about microorganism death, they are dead once they cannot replicate anymore. So microorganism death equals irreversible loss of ability to reproduce. And this is true with antibiotics as well. If they cannot reproduce no matter what, Well, that's when we call it. All right. So there are terms associated with microbial control. Some of these you have heard, some of these maybe not. Maybe some of these you have confused. So I hope to end the confusion. Sterilization. Sterilization is killing or removal of all microorganisms. We set the bar at killing endospores. So, if we want to know if a chemical or an autoclave or something will sterilize, then we ask the question, well, would it kill endospores? Like Clostridium difficile has endospores. Uh, Clostridium botulinum causes botulism. Would it kill those endospores? If it kills those endospores, then yeah, hey, it's sterilized. The procedures and the criteria that we use for sterilization, though, does not consider those prions. Yeah, those are weird. Incineration will work. Autoclaving to a certain extent, chlorine and autoclaving, yeah, but that sometimes causes its own problems. Okay. So, sterilization is not disinfection. Disinfection is the elimination of most or all pathogens. Okay. So, it's not getting rid of or killing everything. When we hear the word disinfectant, we're talking about probably a chemical that is used to disinfect inanimate objects. Sometimes we call disinfectants biocides, germicides, bactericides. More in a bit about those. Okay, antiseptics, these are like really mild disinfectants, but they're different in that we can use these on living tissue. We don't say to little Johnny, oh, you scraped your knee. Let's put a little disinfectant on there. No. Okay. Pasteurization, back to Louis Pasteur. So this is something that reduces bacterial numbers, hopefully gets rid of pathogens, and is used on foods and inanimate objects. More later. Some more terms. Decontamination. Decontamination is the reduction of pathogens to the levels that are considered safe. So that's just going to eliminate the ones we know that cause disease. The types of organisms that don't cause disease, well, they can still be there. Sanitized. Well, it used to be you'd go into a hotel or a motel and they'd have a little piece of paper around the toilet seat that would say sanitized for your health and convenience. I never believed it for a moment. What does it mean exactly? Well, sanitized is supposed to mean substantially reduced microbial population levels. But really, there's not a real number there. There's not a specific level of control. Usually, state and federal health department agencies come in and they set standards and they say, okay, you can only have this many bacteria and you can't have any E. coli there because that means you have fecal contamination, so forth and so on. Preservation. Well, that means that you're talking about the process of delaying food spoilage and other perishable products. So, what would cause food spoilage in, in the first place? Bacteria and fungi are major decomposers. So, a preservative has to inhibit the growth of these types of organisms, growth inhibiting preservatives. Some preservatives are antifungals, some are antibacterials, but and a lot of them are a little bit of both. So keep in mind, sometimes we're just interested in reduce the numbers. You don't always need a blowtorch when a match will do. So, there are certain circumstances when you want sterilization and sometimes when you don't. So, another term, asepsis. Asepsis is the absence of significant contamination. So again, reduce. There's aseptic techniques that are done in the laboratories. microbiology laboratories, pharmaceutical laboratories, and probably what you do in probably in the nurse's station when you're working with when you're working with meds. Aseptic packaging, this is something used in the food industry. So, and there you see it there. Again, so do you always need a blowtorch when a match will do? Well, sometimes yes, sometimes no. So nothing is perfect in all situations. Microbial control methods will depend on the situation. That's true across the board. So, first of all, read the label. When you see a chemical and it says cidal, cide means that it's an agent that kills. Bactericide or bactericidal kills bacteria. Viricide or virucidal kills viruses. Biocide or germicide is being kind of vague. Really, if it says cidal, you need to, even if you've looked at the label, you might want to go on the website and see what it doesn't cover. Chances are they left out the acid fast Mycobacterium tuberculosis. And I'm reasonably sure they would have left out anything that would have killed spores or sporicide. If it says sporicide, you're talking about a very powerful and probably very expensive chemical. So again, biocides, germicides, they usually don't kill endospores. They usually aren't even going to kill the tuberculosis bacterium. So fungicide, well, like bactericide and viricide, this is specifically against fungi. Cidal is to kill. Like homicidal maniac, okay. Homicidal clown, okay. Not into Pennywise, me neither. Okay, well, actually I am, but he freaks me out. Okay, if you see the word static on a label, then the agent is more of a preservative. It prevents growth. Bacteriostatic, fungistatic, this is just going to prevent growth from occurring. So why do that? Well, again, it's a preservative, not only for food, but also on makeup and anything else that is liquidy, that has a shelf life. Probably you want to put something in there that's a preservative. So just listen to this one for a moment, okay? Washing and scrubbing with soaps and detergents achieves routine control. And it's not sexy, but let me tell you, it is the best way to go. You are not going to sterilize your hands. You are not going to get endospores and all the endospores and tuberculosis to die by adding some chemical. So, soap aids in the mechanical removal of organisms. You wash them down the sink. Now, the other good thing is because your skin is your first line of defense, you don't want to destroy your skin while trying to get the germs down the drain, or trying to kill the germs. You want to get rid of the germs without hurting yourself. So, washing with soap and water leaves the beneficial skin microbiota alone because they're going to reside in deeper and underlying layers of skin and hair follicles. So soap, not sexy, but very good. Okay. And yeah, you're not hurt by regular use, believe it or not. Okay, so again, hand washing with soap and water is probably the most important step in stopping the spread of infectious diseases. Of all the things that I am teaching you in this course, this is the most important take-home message because every day you can save lives just by washing your hands. I know. Doesn't seem possible, but this is important. And I know I've mentioned that before, but I keep hitting it. All right. Reduce, reduce, reduce. In the hospital now, okay, sometimes you have to take it up a few levels. You need to minimize the microbial population. That is very important because there are many healthcare associated infections. Here's a wound. This person has Pseudomonas aeruginosa, a gram-negative bacterium that is really nasty, and you don't want it in a surgical ward or a burn ward. Okay. In a hospital, pathogens are more numerous, varied, and they probably have a lot of good genes good genes for them, so they're very virulent. They're probably a little more virulent in the hospitals. The hosts, again, are weakened, so the immune system isn't working. So this is perfect for them. All defenses are lowered, so the germs move in. And again, if you're going in for a surgical procedure, minor or major, well, your first line of defense, is breached. So all your barriers like skin, all of that, okay, is now open for infection. So let's talk about how microorganisms die. They actually die really fast at first, but trying to get the last ones to die is awfully hard. And I mean, not only in a toilet bowl, but also in the body. So this will come up a little bit later on. So note, just picture, if you will, a filthy toilet. You know what I'm talking about. There's a biofilm ring and it's slimy and it's awful. So, if you spray that toilet bowl with some Lysol, okay. So not all of the microorganisms are going to die at once. If you think of these chemicals like chemical bullets, well, you're going to hit the ones on the top first, second, and you're going to keep hitting them over and over again, instead of, again, little chemical bullets. The ones on the bottom, close to the porcelain, they're all protected. So with chemicals, and we will talk about chemical agents, you need to physically remove these organisms by chemical washing and scrubbing. Yeah, I know people hate scrubbing their toilet, and I'm sorry, scrubbing bubbles is not going to cut it. Sorry, I'm just being truthful. Okay, so mechanical washing and scrubbing, and then you can once you scrub, then you can add the chemicals. Okay, and that will work much better. Reduce, reduce, reduce. You must do this. Okay. All right. So, microbe susceptibility. Are all microorganisms created equally? Well, you've already learned there are some that are tougher than others. Not all microbes have the same susceptibility to disinfectants, antiseptics, and antibiotics. So we look at things, okay, is it a gram positive, gram negative? Does it have an envelope? Is it a virus? Is it an envelope? Does it have mycolic acid, endospores? Well, here's the list, everybody. So if you have a prion, this kind of rules at the top, to be honest. There's no prions here, but it would be at the very top. So the hardest thing to kill, and what sets the bar for sterilization, endospores. When a bacterial cell goes into the endospore phase of its life, it is the toughest living thing to kill on this planet. And I can say that because prions aren't living, so there you are. Okay, and then what's next? Mycobacteria, tuberculosis, everybody. We haven't talked about protozoans, but protozoans, if you've heard of Giardia, yeah, chlorine isn't going to work on this. And actually, protozoans are pretty tough all around, so they're kind of in the middle. Gram-negative bacteria, they have that outer extra membrane. Now, it's not the mycolic acid that tuberculosis has, but it's still a protective layer. Fungi, a little easier. And then we have our viruses. Remember, viruses without envelopes are tougher to kill in the environment, okay? And gram positives with their cell wall sticking out there with no protective layers. There's not an outer freakish membrane. There's not any mycolic acid. Yeah, they're pretty much the, the biggest victims of the bacterial world. When it comes to all of these, though, viruses with lipid envelopes. You're like, well, hey, but keep in mind, HIV has a lipid envelope. So does influenza. Influenza travels in the air. You're not going to be able to disinfect those droplets. So, yeah, it's only a lot of this is only going to be relevant on hard surfaces. Biofilm. Here we are. There's a catheter. Remember, biofilms require extra considerations. Here's some biofilm with bacteria and yeast. And again, the outer layers that are secreted, that's going to protect them. And it makes these organisms sometimes up to 1,000 times tougher to kill, right? And by the way, Pseudomonas aeruginosa is really good at creating biofilms, okay? Okay, so that was a lot of general information, but okay, let's apply this. What can I do to control microbial growth? As I said, we're going to talk about physical means of control and chemical means of control. Let's start with the physical methods. A lot of these you may not need, you might be aware of all of this in the background. But there are a few of you. If you're a surgical nurse, this is probably pretty important to you. So first of all, using physical control methods, controlling microorganisms with high temperatures. So here we have, yes, something boiling, whoops, on the stove. High temperatures, is that going to work? Well, it depends. How much? Well, that's going to vary. What do you mean? Well, okay, high temperatures alone tend not to penetrate every little crevice, so heat with moisture is always best. So just at home, a little hint. If you use a sponge to clean your counters, that sponge is going to be extremely porous and lots of very, very, very microscopic little areas for bacteria to hide, and viruses too. It's going to be moist, and because you're picking up food bits, it's going to have lots of food there. So microorganisms are going to love your sponge. So the best way to do something about that is to get a bowl, fill it with water, put the sponge in the bowl with water, put everything in the microwave oven, and heat it up. The water will eventually boil and the hot steam will penetrate the sponge, and that will prolong the life of your sponge. So if you're a sponge person, that's the one way to do this. So heat with moisture is always better. Okay, steam a lot of times can be much hotter than the water, but there are some so increasing steam in water increases the efficiency of heat killing, plain and simple. So it can kill vegetative microorganisms and most viruses. So that's good. Is it going to kill endospores? Not reliably. Is it going to kill gram-negative endotoxins like LPS? Yeah, no. Usually incineration is for that. So what we tend to do, and I'm sorry this all came up at once, we tend to add pressure as well as heat and water. Pressure will increase how high the temperature will boil. So what are these numbers, 92 versus 100? If you live in this area in Nebraska or Iowa or around here, you are at sea level. And water will boil at 100 degrees Celsius, getting pretty hot. But if you go to Denver, you can boil and boil and boil all day long. But yeah, because of the difference in pressure, you're only going to get your temperature up to 92 degrees Celsius. So it's not reliable. Boiling like this is not reliable. So again, we can add pressure, and that's really a good physical means of control. In fact, adding pressure makes us sure that we can sterilize, okay? So sterilization using pressurized steam, using pressurized steam, well, he's part of a team. Okay, pressurized steam is best of all. So the autoclave, you could see this gentleman is placing surgical instruments into the autoclave, and there will be water that's pumped in here and heat, but there will also be a lot of pressure. And the pressure, again, is going to raise the temperature that water boils. and it's going to kill the endospores. Absolutely. So typically you can get the temperatures beyond 100 degrees Celsius, all the way up to 121 degrees Celsius, as long as you have enough pressure, like 15 psi. And how long just depends on what you're putting in here. 15 minutes sometimes does it. Yeah, it's clean. Prions, by the way, are thought to be destroyed at 132 degrees Celsius for an hour. What they recommend funeral directors who cannot use, that have trouble, um, they suggest for funeral directors to soak things possibly contaminated with prions in hypochlorite bleach, Clorox, and then autoclave it. The problem is you can't have hypochlorite around the formaldehydes. It's embalming fluid. But anyway, I digress. What else can you use? Pasteurization. So really quickly, when you pasteurize a beverage or milk, it is not sterilized. You can have your container unopened for weeks and months. Nothing was allowed to get into it. But when you pour it out, huge chunks of like cottage cheese will come out. And that's because bacteria were fermenting the milk. And if you looked under the microscope, you would find a lot of bacteria in that milk. In fact, if you looked in here, you'd probably find some bacteria in, I think this is tomato juice. So pasteurization would be used for that. The reason pasteurization is controlled heating at temperatures below boiling, and it is not designed to sterilize, it sanitizes. The reason we don't want to sterilize is because sometimes if you get foods too hot, it will change the consistency, even the flavor, or break down chemicals. So the whole idea is just to reduce the number of microorganisms, reduce, and decrease or eliminate known pathogens. By the way, this is Honey Creek Creamery, and in Council Bluffs, close to Council Bluffs, they have this goat herd, and I had a tour of her facility at some time. So the goats produce the milk, and the goat cheese is so fantastic. It's like cream cheese. You wouldn't know it was even from a goat. It's so good. But yes, so the pasteurization machine is over here. Okay, and that's all she needs to do. Just get it high enough so the the microbial count is down. How about dry heat? Well, it's going to take longer, and if you have plastics or rubbery items, it's going to melt. So that's not going to work very well. Incineration is sometimes used. You can use a flame or electric heating coil, and that will ignite and reduce microbes and also other substances there. This gentleman is incinerating carcasses, and fire will do a fairly good job, but you have to be careful with carcasses because the water inside the carcass, because we're all made up of water, it will expand and explode. So really what he is doing is dangerous. You want to do incineration in an enclosed environment. Okay, like in hospitals, that's what they do. They have an incinerator, it's enclosed. No explosions. Okay. How about using low temperatures? No. Of all the things I'm going to talk about, low temperatures will only kill parasitic worms, and it will do the trick, which is why sushi in this country, is pretty darn safe. You don't have to worry about parasitic worms because of the freezing process that we take the fish through. But worms are animals, so yeah, low temperatures would kill them just like it would kill us for the most part. So, in fact, low temperatures will preserve things. We keep cultures in very low temperatures. This is an ultra low freezer. So, no, Filtration. Yes, you will come upon rooms, the negative pressure rooms that are used very often. This is to make sure that the air does not creep out from one area to another, and particles could contain tuberculosis, bacteria, and viruses. So, yes, a lot of containment facilities will have a negative pressure room. And the air then is taken through and filtered using a very small little uh, uh, kind of filter. The little holes, the pores would be very small in what are called HEPA filters. Okay. Radiation, how about that? Well, there's a lot of radiation. You have to understand, not when we say radiation, we're not talking about the kind of radiation that's going to make you glow in the dark for the rest of your life. Uh, we can expose things to radioactivity and not worry about getting cancer from it. Understand that there's electromagnetic radiation includes radio waves, microwaves, things like that. So, and high frequency has more energy than low frequency. So a radio wave, microwave, infrared, yeah, pretty long wavelengths, not a lot of energy. Doo-doo, doo-doo, doo-doo. But then when you get to ultraviolet, and there's a germicidal kind of ultraviolet, makes the peaks come up faster, increasing wavelength. Yeah, then you get, you also get X-rays and gamma rays and things like that. So gamma rays, X-rays, cathode rays will damage DNA. It will also react with oxygen gas to produce reactive oxygen species like superoxide ions. Nasty little thing there. Okay, and sometimes this is used to sterilize medical supplies and food products. Yeah. Ultraviolet light produces thymine dimers. We already talked about that. It causes DNA to loop together. That will cause a mutation. And by the way, you don't probably have to, I'll tell you what, I'll give you a little hint. All I really care about here is that radiation, not all of it is going to make you radioactive. Know that ultraviolet light is commonly used, and it's very useful for sterilizing. You get these ultraviolet lamps that will uh, help to disinfect counting rooms and pharmaceutical counting rooms and other things. So I just wanted to mention this here that, yeah, radiation will work. And keep in mind, microwaves are also a type of electromagnetic radiation. Just so you know, because of the long wavelengths of microwaves, don't think you can put something in the microwave and sterilize it because the waves will miss spots. You know how food doesn't get heated thoroughly? So you have to rotate the food in the microwave, and even so, not everything gets heated up thoroughly. So don't use microwaves carelessly if you're trying to think, well, I really want to heat this through and through, make it safe. Well, you better cook it a lot, is all I'm saying. All right, let's talk about chemical methods of microbial control. I have Lysol here, not because I am super fond of Lysol products, but this actually has a purpose. I'm going to begin this chemical methods of control lecture and end this part with Lysol products. I wanted to let you know that just because it says Lysol or Clorox, it doesn't tell you what the active ingredient is. You need to start reading labels. All right. So, a little bit about chemicals that attack cell structures. That's what we want to do when we're using these chemicals. We want to hurt and control the germs, like these guys that are making their gooey biofilm. We want to hurt them, but we want to try to do it without hurting our own human cells. See, remember your skin, first line of defense, you need to treat it nicely. So the skin is what keeps out the microbes and keeps out a microbial attack. Again, chemicals is just what you use is going to depend. Okay. And again, no single chemical is best for all purposes. Generally, chemicals are unreliable for sterilization, but I'm going to talk about the ones that do sterilize. They tend to be best at disinfection and preservation. Most chemical agents are inactivated by organic material, biofilms, or goo. See the shower? Blech, yeah. So you got to get in there and scrub. So scrub surfaces first because microbes can be trapped and protected, not only in biofilm, but also blood, pus, and excrement. Oh joy. The first chemical that I'm going to talk about will sterilize. This is called ethylene oxide. It is a gas sterilizer. It has to be used in a vault like this because it's explosive and toxic. But in hospitals, a lot of hospitals have these things. They don't use them that often because it does require, and you don't need to know this for a test, but it requires something from 3 to 12 hours of, sterilization and then 12 hours just to detoxify everything. So, but if you were trying to sterilize plastics and tubes from respiratory therapy, or maybe you had a wheelchair and you have all these little inner places that need to be sterilized because your patient had active TB. Well, then you just have to wheel it in the ethylene oxide gas sterilization vault. And there you go. So it penetrates really well, gets into all the crevices, and it kills spores. That means it sterilizes. Okay? Kills the endospores. So we're good. Yes. So does it sterilize? Yes. Ah, Lysol. Oh, but this isn't every type of Lysol. Okay, this is the special brown bottle Lysol. My mom used to use this, and boy, does it stink. You can't find it everywhere, but because most people can't stand the smell. But this Lysol contained a chemical known as a phenol. Phenols are effective disinfectants. They are known to hurt cell walls, membranes, and proteins. No, you don't need to know that for a test. But if I said, okay, what is the active ingredient in the brown bottle of Lysol? Well, it has phenols or Lysol phenolic disinfectant. Okay, so that's one thing. Now, you may not be familiar with this, but sometimes I'm going to guess you might come in contact with something like this. This is hexachlorophene soap. Phenol. There's a little hint there. Usually it comes combined with detergents. It's pretty good against fungi. Okay, as well as the easier to kill bacteria and viruses. The problem is, though, is that it does damage membranes. So, okay, yes, bacteria have membranes, fungi have membranes. Dang, we have membranes. So it can lead to toxicity problems. If you use this as a surgical scrub, well, okay, you're scrubbing this pretty hard into the tissue. By the way, phenols. Phenols are endocrine disruptors. Yeah, so, okay, endocrine, those are your hormones. Ladies, do you know what happens when your hormones are just slightly off balance? And your hormones are released in tiny, teeny bits and pieces. Okay, very balanced. If they even get slightly unbalanced, chaos occurs. And I'm not just talking about mood. If you are pregnant and you have a developing fetus, and that fetus is trying to develop into a male. Whoa. Oh, shucks. Okay, sorry. So how about another phenol? You're thinking, well, I don't use the brown bottle stuff and I don't use hexachlorophene soap. Well, okay, so there's also a chemical called triclosan. Looking at the name triclosan, you could not tell that it is a phenol, but it is. Was very common in, well, everything that said antibacterial. Hand soaps, hand sanitizers, oh, you name it, it was in everything. Again, phenols mess with membranes. Yeah, germs have membranes, so do we. It can cause hypersensitivity. It's easily absorbed through the skin. It's been found both in human breast milk and urine. And in lab animals, we know that it is an endocrine disruptor, changing the balance of estrogen. And in lab animals, it has shown fetal development problems. But again, in lab animals, because we don't do this in humans. Okay. So if you've ever had something like this, the B&B Bath and Body antibacterial lotion smells really good. They took a lot of this out of their products. I don't think anybody knew the difference anyway, really. But it had 0.3% triclosan in there. Okay. Now, sometimes triclosan is good. It is a sometimes antibacterial. So, let's say you are colonized by methicillin-resistant Staphylococcus aureus, and you've got to get it off. You've got to decolonize yourself, or your patient, or you're prepping before a surgery. Okay, let's take this up to about a 2% solution. And yeah, then we can use triclosan, assuming that you don't have problems, assuming you're not pregnant. I wouldn't do it if you were pregnant. But, then again, I don't know. Okay, what else can we use? Well, there's also a chemical that is not a phenol. This is known as the chlorhexidine disinfectant. So this are not disinfectant, excuse me, antimicrobial. It is common in surgical scrubs. Again, maybe you're familiar with this one. Hibiclens has chlorhexidine in it. That's the one that I always end up using before surgery. Again, membrane disruptor. Combines with detergents or alcohols. Good with some bacteria and fungi and enveloped wimpy envelope or viruses. And it works by denaturing proteins. You don't need to know that, but that's what happens. Doesn't work on endospores, doesn't work on tuberculosis, so mycobacteria, doesn't work on protozoans, doesn't work on non-envelope viruses. So will it sterilize? No, but you're going to be rubbing this into your tissue and bathing in this from head to toe. So this is Hibiclens is a good example, but this would probably work with gram-positive bacteria and envelope viruses because they're pretty wimpy. Okay. Alcohol. Oh, joy. Let's do that. Oops. Nope. Doesn't sterilize. Nope. In spite of what you think, it doesn't. Because it doesn't even touch an endospore. You might as well use harsh language. Just, Anthrax endospores have been reported to survive at least 20 years in alcohol, and the most effective concentration of alcohol is 70%. So even at its best, they can live 20 years and they don't care. So again, you might as well use harsh language, save your money. Again, with alcohols, 70% is generally used. Isopropyl is the rubbing alcohol that we tend to use, but that it does have its uses. You can you can rub it on your skin, but you're using mechanical scrubbing, and you can wipe off thermometers and other pieces of equipment. So you literally physically wipe them off, and it will kill, maybe it might help to kill some of the gram-positive bacteria, and it dries and doesn't leave a residue. So it has its uses. One drawback, though, for people who don't quite get this message and they get a big open wound and they pour alcohol on it, especially in really bad Westerns. Do not do that because alcohols denature proteins and lipids, and they all clump up and form a nice little safe barrier for bacteria to grow down underneath it, especially the anaerobic bacteria that don't like oxygen. Those with the first name Clostridium, like Clostridium perfringens, that causes gas gangrene. If you do this on a big gaping wound, you're going to counter debridement. You know how you open up the tissue, right? So this will counteract everything that you are doing. Do not use alcohol. A little puncture wound is fine, but on a huge open gash, no. So if you write a novel and somebody's trying to clean up a wound, please don't use alcohol. Okay, and then there are alcohol-based hand sanitizers. Now wait a minute. We know those work. Purell spends millions upon millions of dollars putting their product everywhere, and boy, it is. Alcohol-based. Is this going to work even slightly? Not on endospores, not on C. diff, Not on tuberculosis, not on protozoans, not on gram-negative bacteria, not on fungi, probably not on non-envelope viruses, maybe envelope viruses and gram-positives on a really good day. But you know what? Purell is alcohol and goo. That's what hand sanitizers are. So, goo, really? So if you have to use Purell, you're in a pinch, wipe it on your hand and then wipe physically everything on like a towel or something. At least that might help a little bit. What is better? Yeah, nice soap and water down a drain. Yes. Okay. Hydrogen peroxide, this stuff. Hydrogen peroxide is in a brown bottle because it is very reactive. If you leave this open, expose it to air or add water or something like that, it'll break down. Hydrogen peroxide, I don't know if you know the chemical formula, but it's H2O2. So, okay, little extra oxygen there. Now, what happens if this is exposed to air? It breaks down and breaks apart and becomes superoxide ions and hydroxide ions. And these are free radicals, and these are very powerful. It will damage membrane proteins and DNA. As an antiseptic in low concentrations, well, excuse me, at low concentrations, it makes a pretty good antiseptic, and that's what this is. But if you use a strong concentration or a high concentration, it will kill endospores. So will that sterilize? Yes, at strong or concentrated solution at a high concentration, it kills endospores. That means it'll sterilize. You don't use this form though. This is not the form. The form that is very powerful and kills endospores is very toxic and might kill you. So usually it is put into a container, some machine that pumps it through. If you were making contact lenses and you wanted to sterilize them, you might run it through this. So yeah, hydrogen peroxide. It will kill endospores, but only at a very high concentration. Aha, these look familiar. Halogens. Halogens will sterilize slowly. Not really reliably, but listen, you're going to be exposing your cells and all kinds of things to this. But if you use it correctly, it will kill endospores. Okay, but again, it's not that reliable. Iodine, betadine, all of those. This is one of the oldest and most effective antiseptics that we use. But again, it can sterilize. It's used to disinfect skin and even sanitize food utensils. Oh yeah. If you know of a home brewer, they use ionophore, and that they use that to clean their tubes and stuff like that for making wine and beer. It works by oxidation. It knocks the electrons off of all kinds of molecules and will kill germs. But again, it's a sometimes chemical. It can be toxic to human tissue as well. Chlorine, okay, one of the most widely used kinds of chemicals, okay, is used in water treatment, dairy equipment, swimming pools. Works like iodine, but it's not as stable, okay, but still not as toxic either. Now, when you get this bottle, like iodine, usually light cannot penetrate the bottle. That's the way the manufacturers set this up. Why? And why does it tell you to dilute? Why do you need to dilute this? Like a 1 to 10. My mom used to think, well, if it's a good chemical, I'll just use it right out of the bottle. Is it because the manufacturer is trying to just give you a concentrate so that'll save on shelf space? Well, it does do that, but no, the reason you don't add water is because if you added water in the factory, the Clorox would start to break down and become very unstable. Okay, so you can't add it in the factory. Now, Why not add it, why add it when you get home? Why add water when you get home? Well, when you add water to Clorox or sodium hypochlorite, it actually makes the Clorox more acidic. Acidic. It lowers the pH. Is that going to work on microbial control and kill the germs? Oh, you bet. But again, okay, some of this stuff can be very toxic to animal tissue, mucous membrane, so you have to be careful. Aldehydes. Probably the only aldehyde that you might possibly come in contact with or know about is formaldehyde. In nursing school years ago, you would have sheep hearts soaking in formaldehyde, and you didn't need to refrigerate the sheep hearts because there was nothing in there that was going to spoil or rot the sheep hearts. The formaldehyde would kill everything, and it does. Aldehydes will kill spores, the vegetative reproducing cells. Yeah. So, does it sterilize? Oh, you bet, except when it comes to prions. It actually makes things stickier. So aldehydes are used. Now, this is one of the giant squids that the Smithsonian Museum had, and they preserve them in formaldehyde, but they have to be very careful. Once again, the main component of embalming fluid is formaldehyde. You need to have something that's going to kill fungi and bacteria that would cause decomposition. And FYI, it denatures proteins. Seriously. Heavy metals, I'm going to do this both on the same slide. Sometimes you will use creams that have silver in them. And yes, mercury, silver, and copper are antimicrobial naturally. Now, mercury, you're not going to mess with that because it's a neurotoxicant. However, silver and copper, copper pipes, copper like teapots, things like that, all of those just naturally inhibit microbial growth. Silver is commonly used as an antiseptic in creams and bandages. It's not too harsh, but it does the job. Okay, so when it comes to the old money, it's actually not too bad. You think when kids are, have these things in their mouth, like, oh, they're dirty. Well, maybe if there's a biofilm, but it's cleaner than the paper bills. Okay. What else can you do? There's also preservation by reducing water availability. So, okay, throw a bacterial cell in some salt or sugar, something that is very hypertonic, and all the water is going to rush out. Okay, we call this plasmolysis. Remember when you learned about hypotonic, hypertonic? Hypertonic is very salty, okay, or has a lot of solute. Yeah, so you can salt food, dry it out, put salt there, put sugar there. All of that will keep bacteria from growing and reproducing. So again, this is why when we have beef jerky, we dry it, we add a lot of salt, and yeah, so that makes it such a inhospitable hypertonic environment. Bacteria and not even fungi are all that crazy about all that salt. So, and again, causes cellular plasmolysis as the water exits the bacterial cell. How about using acids and alkalis? Well, alkalis are going to be very powerful, but you'd never use that on your skin. Um, so oven cleaner would be a very strong alkali. But I know that there is a trend to use acids for as kind of a cleaning agent. Well, and they're fine. Again, sometimes you don't need a blowtorch. A like a some kind of vinegar solution is fine, is great for just cleaning off countertops and maybe freshening up your refrigerator. But is that going to sterilize? No. Okay. Some of you may have observed the flushing of a suprapubic catheter. This young lady has a hole in her first line of defense, her skin, and the catheter is running through there to her bladder. This catheter is going to be full of bacteria, and bacteria are trying to get in. So one of the things that the RN or the caretaker or she has to do is flush the tube out with a dilute kind of acid. So kind of your acetic acid vinegar like solution. Are you going to have this are you going to have a strong acidic solution? Well, no, that's going in her bladder, so that's not going to work. Okay. And here's the process. And if you've ever done this, it's freaky the first time. Just I've done it because my brother was a quadriplegic, and he's so cool. He's like, just go for it, Sharon. So yeah, you just go for it. But you need to be very gentle. And again, the tissue's very sensitive. You do it carefully, thoroughly, and gently. Okay, take your time. You can do it. Here's a little bit of fun. You don't need to know this, but you may have heard of some natural ingredients used as antiseptics. This is true. Now, if you're trying to use these chemicals to destroy bacteria or viruses that are deep into your tissue, yeah, forget it. Think about all the things that go through, how food goes through the digestive processes. And yeah, it's never going to get to a bacterial cell and cause a very effective, uh, 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 a very effective control. It's not going to work. But on your skin, we have known for a while that some chemicals will work, but you have to add these topically. Okay, so papaya fruit, you have to use it fresh, directly on the wound. Now, I don't do this in the hospital, but maybe if you wanted to try at home. Tea tree oil, sometimes people will use honey. I'm not crazy about honey myself, but eucalyptus oil, that is commonly used. And this is from Deborah Ann Riley, who is Associate Professor of Surgery at Nebraska Med Burn Center. Okay, so I began this lecture and I'm going to end this lecture with Lysol. Okay, you ready? This is a real ad for a Lysol product. Held in a web of indifference. Day after heartbreaking day, I was held in an unyielding web, a web spun by my husband's indifference. I couldn't reach him anymore. Was the fault mine? Well, thinking you know about feminine hygiene, yet trusting to now and then care, can make all the difference in married happiness, as my doctor pointed out. He said never to run such careless risks, prescribed Lysol I'm blew it. Prescribed Lysol brand disinfectant for douching, always. Yeah, I know. But I broke through it. Oh, the joy of finding Tom's love and close companionship once more. Believe me, I followed to the letter my doctor's advice on feminine hygiene. Always use Lysol for douching. I wouldn't be satisfied now with salt, soda, or other homemade solutions. Not with Lysol, a proved germ killer that cleanses so gently, yet so thoroughly. It's easy to use, too, and economical. The very best part is Lysol really works. Okay, yeah, I don't know what the active ingredient is, but if you ask around to your grandmother, your grandparents, maybe even aunts, they might tell you, yeah, I kind of remember this. So, anyway, all right, I hope you enjoyed this lecture. You have a good